My name is Susan Hedstrom. I'm the Executive Director for the Foundation for Prader-Willi Research, and I'm really excited to have all of you joining us today as we share the progress that's taking place in PWS research. Joining me today is Theresa Strong, Director of Research Programs for FPWR. Theresa will provide an update on PWS research activities, including recent outcomes from our grants program and details on our most recently funded grants. As always, we do want to hear from you, so if you have any questions as we go through today's presentation, please feel free to submit them through the Q&A. And with that, I'm going to hand it over to Theresa.
Great. Thank you so much, Susan. I'm excited to share today some of the recent progress in PWS research, as well as the new grants that we're funding in the second half of 2022. Oops. So I'll jump right in since we have a fair bit to cover. So first, we'll talk about an update on the PWS research activities, including some of the outcomes from our grant program. And then we'll spend most of the time talking about the new grants that have just been funded, but hopefully we'll have a lot of time at the end for questions and discussions. Just want to remind you of FPWR's mission, which is to eliminate the challenges of PWS through the advancement of research and therapeutic development. And really everything that we do really focuses on advancing that mission. So just a review for many of you, this is a diagram of the pipeline on how we develop new therapies, we being the entire scientific and pharmaceutical industry areas. So we start with research and discovery, identifying those pathways that might be altered in a disease state or in PWS in this case, and identifying what might be a target to drug and to restore things to a more normal state. Once we identify that, we move to preclinical development where we test those drugs or interventions in cell models of the disorder as well as animal models. And then if that looks promising, it moves into clinical development where we start with small clinical trials of phase 1 just to make sure that what we think is going to happen actually happens and that the drug or intervention looks safe. And then increasingly larger trials that are really testing to see whether the intervention is effective in treating whatever aspect of the disorder that we're trying to treat. If we receive FDA approval for the intervention, then the next phase is the post-approval phase, which is trying to use that drug, for example, in the best way possible. Is it better for young kids or older kids or with this disease state or that? So this is the general process that is followed. And our goal at FPWR is to try to accelerate every step of this process. So, support research in drug discovery and but then see how we can move those things forward as quickly and efficiently as possible. And one role that I think is really important for patient advocacy groups such as FPWR to take is to foster, be a place to foster collaboration across different disciplines and across different stakeholder groups. So bring together the patients and the pharmaceutical companies so that the patient voice can be heard, or the pharmaceutical companies with the clinicians so that we can be more efficient in how we do clinical trials. And I'll touch on a couple of examples of how we're doing that. But to facilitate this research and really, again, try to accelerate everything moving from left to right, we've built some tools and resources that can be used by many researchers in the space to help support their work in PWS. And those are shown on the bottom. I'll just mention a couple of them. The Global PWS Registry, which many of you have participated in, which provides information for clinicians to understand PWS, for pharmaceutical companies to understand the needs of the community, and for our own patient advocacy groups to understand the needs of our community. The PWS Clinical Investigation Collaborative is a new group of more than 25 clinical sites across North America, and they're asking questions about how best to care for people with PWS. And then the PWS Clinical Trials Consortium, again, is one of those groups that convenes different stakeholder groups. So there's patient advocacy groups, including FPWR, PWS USA, and IPSO, academic investigators with expertise in PWS, and then pharmaceutical companies that are interested in doing clinical trials. So bringing them all together to address things like what are the best endpoints for clinical trials, how best to do clinical trials in this population and improve efficiency. So that brings me to a couple of industry updates. As might be, it's been very active in the PWS field over the last several years, which is great. There have been some ups and downs, of course. We learned, for example, at the conference earlier, well, I guess it was early last month, that Radius Health, which was planning a clinical trial in PWS, was bought by a venture capital group and closed that trial. So that was disappointing, but there's also been some encouraging news. You may have seen earlier this week, Harmony Biosciences released a press release that they had their top line data from their phase 2 study looking at the efficacy of pitolisant in the PWS population. And they found that data encouraging. And they'll also be looking at the more complete data set, which includes, so the top line data was really looking at sleepiness, but their more complete data set looks at effects on things like mood and cognition, which of course is very important to families. So we're looking forward to additional progress on that and hopefully moving into a phase 3 study. Soleno Therapeutics recently announced that they are initiating their randomized withdrawal study. So you may recall Soleno is advancing DCCR as a potential therapy for hyperphagia and also for behaviors associated with PWS. And they have some very encouraging data. The FDA asked for some more data on efficacy prior to considering whether to approve the drug. And so Soleno and FDA had some discussions, and apparently what they settled on was this randomized withdrawal trial, which we are hopeful will provide the additional evidence, along with some other additional evidence that Soleno has been working on, to advance this to a drug approval. So we continue to look forward to that. Just want to give a shout out as well to our Path for PWS participants who have provided data that has also been supportive of the efficacy of DCCR through the registry. So we're working closely with these companies and doing what we can to help move things forward. In addition, there's several companies that are currently recruiting for clinical trials with new drugs that have potential for PWS or will soon be recruiting. Those are shown on the bottom, a few drug companies, Gedeon Richter, Aardvark Therapeutics, and Consonance, as well as a couple of academic organizations, Montefiore and Maimonides, that also have ongoing clinical trials. So you can find more information about these PWS clinical trials on our website. Of course, deciding to participate in a clinical trial is a very personal decision, but we do encourage you to learn about these clinical trials, consider enrolling. Really, the only way we're going to find out if these therapies are effective for our children is for us to enroll in clinical trials. There's really no other way to develop new therapies. So you can learn more about the clinical trials, and that includes information about, what you might want to ask as you're considering a clinical trial on our website. You might also want to look at our YouTube channel. It has the recordings of the family conference sessions, including the PWS clinical trials panel, which goes through several of the clinical trials that are currently recruiting or will be recruiting. And the link is shown there. So those are some good places for resources for learning more about clinical trials. And just want to mention as well, you may have seen an email, or hopefully you've seen an email or a social media posting about a project that Lauren Schwartz is advancing. And the question here is, we now have a few clinical trials under our belt as a PWS community, and are there ways to improve the clinical trial experience? We, want these trials to be completed efficiently. And those of you who have children or young adults who have participated in a clinical trial probably have some thoughts about how that experience could be made easier for the person with PWS and the caregiver. So we would love to hear those thoughts. So we have posted some information about a survey where we're going to gather data that we can then share back with the pharmaceutical companies or academic centers that are doing clinical trials. Lauren is also interviewing clinical trial sites to get their perspective on how to make clinical trials better. And then we'll be pulling all of that information together and sharing it with the pharmaceutical companies that are in the space. Again, to make the clinical trial experience better for families with PWS so that, again, more families will feel encouraged to enroll. And so that study is, part of the work plan of the Clinical Trial Consortium this year towards improving clinical trials in the PWS space. Also wanted to touch base on a couple of other things that are ongoing that you may not have heard about through other means. You may recall that FPWR has been supporting an effort to advance newborn screening. So we know that most babies with PWS are now diagnosed in the first month or two that they're in the hospital. However, there's still a significant proportion, and we estimate about 15% of babies that don't get diagnosed in that newborn period. And sometimes it takes years, even now, for children with PWS to be diagnosed. And that's obviously a problem. We now know that growth hormone therapy is really better when it's started earlier. Parent education, making sure that the children are getting the right nutrition is also really important. So there's a lot of reasons that we want every baby with PWS to be identified right in that newborn period. And so we supported a study by Dr. Godler, who had a cost-effective method of doing newborn screening that not only picks up PWS, but also picks up Angelman syndrome and DUP15, which are, they're all chromosome 15 disorders in the same region. And that's important because if you're going to do newborn screening, it has to be cost effective. You're talking about screening all babies that are born. And so it has to be an efficient and cost effective process. So that study has gone well, and now we've partnered with some other advocacy organizations, FAST and the Angelman Syndrome Foundation in the Angelman syndrome space, as well as the DUP15 Alliance, to pilot a newborn screen in North Carolina as a first step towards potentially bringing this to nationwide newborn screening. And then in addition, and an example of, what we'd love to see is Dr. Godler, based on the investment that we made and, the initial funding to develop the assay and, validate it, has now got a $3 million grant from the Australian government. We'd love to see that kind of return on investment where we invest it's big for us, but a relatively small amount into a research project. And then that investigator turns around and, is able to get much larger funding. So Dr. Godler will be assessing. He'll be doing 100,000 newborns in Australia and identifying those who have PWS or Angelman or DUP15, and then looking at the outcomes of those individuals. So what is their use of the healthcare system and how do they develop compared to kids that, weren't diagnosed right at birth? And so all of that will provide a basis for or a rationale for potentially having this newborn screen in wider populations. So again, just a few random updates on different aspects of PWS research that are very active these days. One of the things at the conference in Chicago last month that was really notable was several registry-based projects were presented, including the use of dietary supplements. So many of you have been good enough to complete your survey on using dietary supplements, and Dr. Hack and her students are studying that to help understand what supplements are being used and what parents are finding useful. So, is an ongoing project. The preliminary results were presented at the conference. Lisa Mattisbach from FPWR presented some HQCT scores in individuals with PWS compared to typically developing individuals. A lot of you helped us gather that data, and we're preparing a manuscript right now that we'll share shortly. And then finally, Dr. Sani Roy and a team of clinicians as part of the Clinical Investigation Collaborative, have developed a survey on feeding tube use and complications. And their goal is to provide standards of care or recommendations on the use of feeding tubes in babies with PWS. So all of these very diverse aspects of caring for individuals with PWS and doing trials and studies really have come from the information in the registry. So thank you to everybody who continues to add data to the registry. The more data we have, the stronger that information is, and the more likely it is to really represent the entire community, and that's really critical here. I also wanted to mention the PWS Anxiousness and Distress questionnaire, which was just published recently. This arose from a project, a collaboration really between FPWR and Levo Therapeutics. And it really was led by Sarah Cotter, who was the CEO of Levo. She noted that the existing measures of anxiety or anxiousness that are used in clinical trials that the FDA is comfortable with really didn't apply to PWS. So some of the characteristics that we see in PWS, like the repetitive questioning about schedules and anxiety about any change to routine, are really pretty unique to PWS, and we're not being captured by these other instruments. And so we worked with Sarah Cotter and also with Sherry Fennell at RTI, who has a vast amount of experience in generating questionnaires that are compliant with the FDA rules, and there are a lot of FDA rules about developing questionnaires, to develop this PWS Anxiousness and Distress questionnaire. So we used the FDA guidances on patient-reported outcome measures, which included doing qualitative interviews. So some of you did interviews about this instrument. Thank you so much for doing that. It really provided great feedback to improve the instrument. We then did a psychometric validation, and that used the global PWS registry. So about 400 families participated in that, and that provided really robust data for how this instrument behaves in the PWS population. So that was invaluable. And then the PADQ, as we call it, was used in the Levo's CARE PWS clinical trial of carbetocin. And importantly, it showed the sensitivity to change. So you definitely want to see that when you develop an instrument like that, a questionnaire like that, that if there's treatment, you can show an improvement. And this publication has all of that data, which is a ton of data, both on the psychometric validation and then on showing that the questionnaire is good for measuring improvements in these really important aspects of PWS. We know, like the anxiousness and that distressed feeling that people with PWS feel is really, important to them. It's really difficult for them, and it's difficult for the whole family. So Levo has donated this instrument to FPWR, and that's fantastic because now we can share it with both clinical sites, academic sites, and pharmaceutical companies that have treatments, either drugs or non-drug treatments that might reduce anxiousness in our kids. So we're very excited to see that. It was a long process, but it's nice to see it added to our toolbox of clinical outcome measures that can be used for assessing these aspects of PWS that are so important to families. Okay, so now I'm going to move on to our grant program. That's just, again, a review of some of the things that are new in the last couple of months in PWS research. But now I'll talk about the grants that we have funded. The grant program really has been the core way that we have supported research in the PWS community over the last almost 20 years now. We've supported more than 200 research projects, and that's resulted in publications on everything from better understanding the underlying basis of PWS all the way through to doing initial clinical trials. So the first DCCR trial was performed with FPWR funding, was co-funded by FPWR. And optimization of clinical care in PWS. And again, we have a number of examples, some more that we'll share soon about how starting a research program, so giving that initial amount of funding has really turned around and allowed those researchers to get a whole lot more funding to expand their work and efficiently address some of the large projects in PWS. And it's also allowed us to build the resources, cell lines, and animal models that can be used by many researchers, again, kind of lifting the whole field. And I don't have time to go through all of the things that have come out of that grant program. I would suggest, if you're interested, again, on our YouTube channel of the 2022 family conference sessions, there's a research roundup, which is a summary of the FPWR research conference. At that conference, again, which was at the end of September to October 1st, there were about 30 oral presentations, 20 poster presentations, 100 scientists, presenters from 8 different countries. So it was really an international event. We had a lot of young, smart investigators, which was really great to see. So we had travel awards for the students and postdoctoral fellows. We had some small special interest meetings, for example, on, just Magel-2 biology. And there were lots of discussions, interactions, and many people saying, I have a list of experiments to do when I get home, which is exactly what we want to see. So a summary of many of the findings that were presented there is available again on our YouTube channel. And I would just say, from a personal perspective, it's been really gratifying to see the field mature, where we now have a really, I think, because FPWR has been able to provide a steady source of funding for the field, we have a really great critical mass of researchers who are coming together and sharing ideas and moving things forward. I did want to mention one recent outcome that I thought might be of particular interest, and that is a study by the group at Ohio State, Dr. Cow and her colleagues. And that is really looking at one of the targets that we've talked about for a couple of years now, BDNF, which is a brain-derived neurotropic factor. So it's like a growth factor for neurons in your brain. And it's been shown that in PWS, for reasons that are not entirely clear yet, but there's decreased levels of BDNF. This group had previously shown in other models of obesity, so just sort of common obesity, that if you increase the level of BDNF, that those animals, in animal studies, see a decrease in obesity, an increase in lean mass, and better metabolism. And so she was very interested in trying this in PWS. And so they used one of the PWS mouse models and, did show that there was a real change in the metabolism of those animals. So their lean mass increased, their insulin sensitivity and glucose metabolism improved, their energy expenditure improved. And also, which was really encouraging, some of their social behaviors, which are, are problematic, as well as their cognitive ability, which has shown some deficits, were corrected. So not only does this seem to be improving metabolism, but it also seems to have a positive impact on cognition and social behaviors, all of which obviously are really important for our children. So, through through all the research that has been done, we have many targets for therapy now. And I think one of our tasks as a field is really to prioritize those. And studies like these help us prioritize, a molecule such as BDNF as potentially one that we want to move to the top of the line. Okay, so I'm going to jump now into the new grants that are funded. And again, this, it's a smattering of studies to understand PWS better, identify new targets, and then test whether those targets are really, should be prioritized above other targets that have been identified. So I'll break this up into 3 groups of grants and go through them, and maybe pause for questions in between so that you don't get tired of listening to me just run through them. So the first set is really focused on foundational knowledge, so advancing our understanding of the genetics and cell biology of PWS. And there are 3 projects in this group. But all 3 are really addressing that question of how does the loss of these PWS genes, and those are the genes that are in blue, how does that change cell function and lead to the characteristics of PWS? And how can we use that knowledge then to identify new therapies? So that's the big picture. And the first grant in this group is a grant from David Tollerby looking at the non-coding RNAs in the PWS region. So Dr. Tollerby is an expert in these SNO RNAs. So the PWS region includes these unusual RNAs that don't make a protein. They presumably go around and regulate other RNAs. And that in part is why PWS is so complex to really understand. But Dr. Tollerby's expertise is in understanding the role of SNO RNAs. So there are SNO RNAs in the PWS locus, but there are other SNO RNAs as well. And that's what his career has been focused on, is really understanding what those SNO RNAs do. So this project is going to use all of that expertise and the advanced technology that his group in many cases has developed to better understand what our SNO RNAs are doing. So how are they interacting with other proteins? How are they interacting with other RNAs? And what happens when they're lost? So we're excited about this project because this is a very experienced researcher in SNO RNAs. He's really sort of a giant in the field of SNO RNAs, and that their lab uses some state-of-the-art technology. So it's not simple to look at how SNO RNAs work. So not everybody can do it, and this lab has a lot of expertise in that. And the study may provide insight into the underlying molecular basis of PWS, and that may suggest new targets for treatment. So the goal is to identify how brain-specific SNO RNAs in the PWS region are regulated and how they function. The next study is from Dr. Jeffrey Friedman at Rockefeller, looking at the cellular and molecular basis for obesity and PWS. And the rationale here is that we know that the hypothalamus is likely important in regulating appetite in PWS, but we don't know the specific cells within the hypothalamus that are kind of driving the deficits in PWS. And there are new technologies that the Friedman Lab is using to identify cell type specific changes in how these cells function. So Dr. Friedman, who is, really, again, another giant in the field of obesity, his group identified leptin, one of the first regulators of appetite known. He's going to use this multiplex single nuclei RNA sequencing, again, a relatively new technique, and apply it to identifying the specific neurons that cause hyperphagia in PWS, and use that information to screen for drugs that might correct these deficits. So this is a newly developed and powerful tool. Data from this study should be useful to many researchers in understanding the differences at a cellular level between the hypothalamus in PWS and the hypothalamus in typical individuals or mice in this case. A very experienced researcher in the obesity field, so really has an understanding of what drugs are out there and might be applicable to PWS. And this group has had success in applying this approach to other obesity models. So the long-term goal here is to identify the underlying cause of hyperphagia and then validate new drug targets that might be useful in PWS. The third study in this group of foundational knowledge is from Dr. Tim Wells at Cardiff University, and this is a renewal. So we provided funding to Dr. Wells and his group maybe 2 years ago, and they used that to look at the morphology or the shape and connections of neurons in the brains of a PWS mouse model. And that's important because it might provide insight into some of the cognitive difficulties that our children have. And they did some really beautiful work, which was presented at the conference, about how the neurons are shaped differently and how their connections are different than a typical brain. And so in year 2, Dr. Wells is going to look more closely at the electrical activity and functional circuits. So how does that alter how the neurons interact? And look at a detailed assessment of how that changes over development and determine the impact of those changes on cognitive flexibility. So we know our kids have a real problem with change and transition from one thing to another, and this could underlie that. So it's a project to really get a grasp on what the differences are so that we can start to think about how we might address those. So cognition is an understudied area in PWS. It's very important, obviously, to all of our families, but really the basis of cognitive deficits in PWS is very much unknown. So this will be a really important step to better understanding these problems. And if we better understand the problems, then we can begin to, look at different therapeutics and see how they would apply to PWS. So there are many genetic disorders now that are looking at therapeutics to improve cognition in other genetic disorders like Fragile X syndrome, Down syndrome, and we need to understand whether PWS, if the same drugs might be useful or not in PWS. So the long-term goal of this project is to address, a major deficit in our understanding. What is the basis of cognitive deficits in PWS, and first in the mouse model and ultimately in people with PWS. So Susan, I'll stop and see if there are any questions on those first few projects or any other questions that have popped up before moving on. Is there anything in the queue?
First, Theresa, thank you so much for telling us more about those projects. They're very deeply embedded in science, and it's hard for a lot of us to understand. But I can say from a personal standpoint that any research into cognition that could improve my son's cognition is welcome research. We have a couple questions in the queue, so I'm going to go ahead and throw those at you. The first one actually goes back to the newborn screening that you were talking about. Will that identify both deletions and PWS by UPD?
Yes, that's a newborn screen that would detect, it's like a methylation test, so it would detect more than 99% of cases of PWS. So deletion, UPD, as well as imprinting center defects all would be picked up by that.
Mm-hmm. Great. And then in several of these studies, you've mentioned that mouse models are being used. Why are there so many studies that are utilizing mouse models?
Well, I mean, I think the simple answer is that we have good mouse models of PWS, and it's easier to look at a mouse brain than it is as a human brain. Obviously, it's important then to relate that to the human situation. And, we do have brain banks that have PWS samples, but of course, those are precious samples because they're, from deceased individuals with PWS. And so, we don't want to sort of use them up willy-nilly. So, using the mouse models to gain an understanding of what questions to ask in the human tissue is really important. And now with iPS cells, we can also complement those studies using essentially a brain in a dish. It's not exactly the same thing, but there is some information that you can get from those. And people are doing that as well. So the mouse model is a good starting place. And then, that those, once you have well-formed questions, you can use some of the other human resources. But obviously we want to look at these things in mice before we move them to humans so that we know that we're looking at the right thing.
Great. Thank you so much, Theresa. I think we're ready to go on to your next group of grants.
Okay. Oops. The next group of grants is identifying new targets for PWS therapies, and those are shown here. The first one in this group is a group in Spain they actually have a really nice group of researchers who are interested in PWS, and one of those researchers, Dr. Castellano, is really interested in understanding the role of fat sensing in altered feeding behavior. So we know that our bodies send signals to our brain about, our nutrient status, and some of those signals are in the form of fat molecules. And so this fat sensing is really important in regulating appetite and body composition. And surprisingly, it really has not been explored very much in PWS. You might wonder if the fat sensing mechanism is awry in PWS because individuals store so much fat, the body doesn't seem to be recognizing that well, but it really hasn't been studied very much. And so Dr. Castellano is going to be looking in PWS mouse models to see if this signaling system is intact. So this is a novel pathway examining potential alterations in lipid sensing in PWS. It may provide opportunities to pursue novel treatment strategies that are based on that fat sensing pathways. And we also are enthusiastic about this study because it is an early stage, productive young scientist. He's in a supportive environment, and it really may provide a critical funding to really build the collaborations and programs that group is developing. So the long-term goal of this study is to assess the contribution of lipid signaling pathways to the metabolic alterations in PWS throughout development, laying the groundwork for the potential for new therapeutics. The next grant in this group is from Dr. O'Connor at Mount Sinai, looking at the orexin system to treat PWS-associated hyperphagia. So we know that loss of one of the PWS genes, MAGEL2, lowers orexin in the brain. So orexin is a neurotransmitter. It's important in wakefulness, but also things like appetite and mood. And the potential of targeting the orexin signal to improve hyperphagia really hasn't been looked at in great depth. And so Dr. O'Connor is going to use brain mapping techniques to look at the circuitry and understand how a reduction in orexin levels might lead to altered brain connectivity and function, and then test whether some of the novel medications that are being developed for orexin signaling might correct those deficits. So we're excited about this study because it's using cutting-edge technology and an innovative approach to look at this particular pathway, which we know is altered in PWS, and which we know, many of the symptoms associated with PWS, sleepiness, hyperphagia, are tied into this pathway. The development of drugs targeting the orexin system is a very active area of investigation, not only for PWS, but for many other disorders. So if this hypothesis is supported, there may be the opportunity to try some new therapeutic options. And we also like this study because it's a very well-trained young investigator just getting his lab going. And, this might be a very nice project for him to really learn about PWS, and we hope stay in the PWS field long-term. So the long-term goal of this project is to enhance our understanding of the changes in the brain that lead to these hunger and behavior problems in PWS, establish the rationale, whether, how much of this is based on changes in the orexin system, and consider new therapeutics based on that. The next study is from Dr. Zhang at Columbia University, and this is looking at SNORD116, which you may be familiar with. It's known to be one of the critical genes in the PWS region. It's one of these SNO RNAs, and she is in, and it's lost in all cases of PWS that have been identified. So this lab, which is the part of the Libel lab at Columbia, has been studying the function of SNORD116 for a number of years, and they've shown that a particular gene called DGKK is a candidate for being affected when SNORD116 is lost. So what they see both in mice and humans, again, and you like to see that connection between the mouse models and the human models, is that loss of SNORD116 increases the expression of this protein, which may then go on and have metabolic consequences. So this project is going to investigate the interaction of SNORD116 with DGKK and look at the downstream impacts when that molecule is abnormally expressed. So we're excited about this study because it's an experienced PWS research lab. This pathway is really interesting. It impacts other pathways that have been shown to be disrupted in PWS, including the mTOR pathway and cyclic AMP levels. And if this molecule of DGKK is implicated as a downstream player, there are already drugs available to modulate its activity. And so it could be a pretty sort of a shortcut to a potential new therapeutic for PWS. So the goal is to identify the role, the long-term goal is to identify the role of SNORD116 in this pathway and suggest a new pharmacological targets. The next study in this group is from Dr. Godler, and this is again is a renewal. So we had provided funding to Dr. Godler in previous cycles to look at what genes are differentially expressed both in blood and brain in individuals with PWS. So this is using human samples, and ask whether those gene expression differences are correlated with some of the behavior problems, the autism, the risk of mental health problems in PWS. And so Dr. Godler is looking at many things. He's comparing blood and brain in PWS compared to normal or typical, but is also comparing deletion to uniparental disomy to gain an understanding of why we see differences between those two subgroups. And he's done a lot of work on that and has, data sets about changes in gene expression across these different groups. And now we'll be, taking that to the next step. So in year 2, they're going to expand their studies to some additional samples. So they've done a number of brain samples, probably the largest group of PWS brain samples that's been investigated. They're going to expand that. And then they're looking in particular, they're interested in this gene UBE3A, which is in the PWS region. It appears to be expressed at higher levels in individuals with UPD and imprinting center mutations. And it appears that the higher it is, the more difficulty those individuals have, for example, with social cognition. So, the study will now expand samples and really, look at it in a larger sample set and correlate it with the severity of some of those behavioral changes. He's also going to use artificial intelligence to query those data sets and identify potential drugs that might improve these aspects of PWS. So we're excited about this study because it's been a very productive project in year 1. He's got very significant data sets. He's very collaborative. So again, it might be information that can be used across researchers and looking at the differences, again, between deletion and UPD. The application of new artificial intelligence approaches to identify drug candidates has been used in other fields, hasn't really been applied to PWS, so we're excited to see that applied here. And this is an experienced PWS researcher. So the goal here is to define the genetic pathways that are dysregulated in PWS blood and brain compared to typical individuals, as well as between the subgroups, and identify existing medications that could be used to correct some of these changes. Next, we have a study from Drs. Chen and Betley. These are two investigators who have worked together for a long time investigating food intake. So the neurobiology of what drives us to eat and what drives us to stop eating. And these investigators have published late last year a really exciting and thought-provoking paper in the journal Nature, which is a big deal in science as an article in Nature. What they found is that an unexpected region of the brain controls satiation. So satiation, or the feeling of fullness, it seems to be that satiation activity seems to originate from this anterior deep cerebellar nuclei. So in the cerebellum rather than, for example, in the hypothalamus. What they found was that they could activate this network, and it would cause animals to stop eating. So they became, they felt full. And they also did some imaging studies in humans with PWS, and they found that this network, this activation, is defective. So that is really changes our thinking about, some of the food behaviors in PWS. Maybe this initiation of satiety or feeling of fullness is really underlying many of the problems that we see. So this opens up a lot of new areas of investigation. There are other projects that are coming down the line in humans, but this one will be in mouse models of PWS now. So seeing if the mouse models of PWS also show more of this deficit, which will allow better screening of drugs. If they can show that this activation of the satiation network is defective in PWS mouse models, then you can do a lot of screening of drugs and find the ones that address this issue best. And that, would be great to be able to do that in a mouse rather than in human clinical trials, which take much longer. So we're excited about this project because it's an innovative project assessing a novel neural network that's important in initiating satiety. And I don't know about you, but my son with PWS, a lot of it seems to be that, he can eat and eat and eat without getting that sense of fullness. So it just makes sense, I think. There's direct relevance to changes in eating patterns in PWS, and this is a group that has worked collaboratively for a long time and really made some strides in understanding feeding behaviors. So this project, we believe, will help guide the development of other projects that may have novel ways of treating the aberrant behaviors, eating behaviors that we see in PWS. So I'll stop. We just have a few more, Susan. I know we're getting short on time. If there's a pressing question, maybe we can take one. Otherwise, I'll just plow through the last few grants.
Sure. Let's just do one. You had mentioned in the orexin study that there's known drugs that work on that pathway that could help speed up making a drug available should orexin be an issue. How much faster is that if you take a drug that's already known versus a drug that needs to be tested in humans for the first time?
I think that shaves years off the drug development pathway. So if it's been in humans, it's shown to be safe, you would then just need to and presumably if it's shown to have efficacy, you would know kind of what your endpoint should be, and you would just need to look at it in PWS. So you could kind of go straight into a pivotal efficacy trial, potentially anyway, rather than having to do all the safety studies in animals, then do the safety studies in humans, and then do the clinical trial. So that's really shaving years off the process.
Sounds like a great way to go.
Right. And that's, for example, why Dr. Godler is also he's using AI to identify existing drugs, because it's much faster to identify those drugs that, exist that we didn't really think would be applicable to PWS. Maybe we're not seeing it, but, with this new technology, we can pick out the drugs that might be really efficacious in PWS. And this is an approach that is being used in other fields. And again, it's really helping things move along a lot more quickly.
Sounds really exciting. Anything that can help speed the process, it sounds like a great way to go. We do need to just keep breezing through these grants, so I'll let you keep going.
Okay, great. Just a couple of more, two that are in the area of genetic therapy, which of course is near and dear to my heart. So I'll go through these quickly. We know that the basic concept is that every person with PWS has the maternal genes, the PWS genes sitting there on the maternal chromosome, whether you're deletion or UPD, that you have one or two maternal chromosomes. The genes are there, they're just inactive. And so the next two grants are really about trying to activate those genes. This first one is from Dr. Butler at UT Southwestern, and he's using a genetic therapy approach to knock down a negative regulator of the gene activation. So it's like a double negative. So this protein, which is sometimes called G9A, sometimes called EHMT2, silences the PWS region on the maternal chromosome. And so he's using an approach to block that so that the genes get unsilenced and become active. We're excited about this study because it builds on some previous work that FPWR funded, Dr. Zhang's work, showing that this molecule is a key regulator of gene expression in the PWS region, but it's a different way of trying to knock that gene down and allow re-expression. So it takes a different approach. And it's really important at this stage, which is still early days for genetic therapy of PWS, to look at different approaches and then try to assess which is the more feasible approach to move into clinical trials in humans with PWS. The next study is also looking at maternal activation of the PWS region genes using a different approach. This is a year 2 study. So this group at Duke University has previously used a CRISPR-based approach to do maternal gene activation in PWS. And they showed that they were able to reactivate the maternal genes in about 20% of PWS cells. And that's one question we have not yet answered as a field, is like, what percent of activation needs to happen in order for there to be benefit? But this group is really focused on, okay, so how do we get that percent up? And then how do we maintain expression in the long term? Because those are questions that are going to need to be addressed if we're going to do genetic therapy. So in year 2, they're going to determine the epigenetic requirements for a uniform and stable reactivation of these genes in human cells using these CRISPR-based approaches. We're excited about this therapy because, like any epigenetic therapy, it has the potential, at least, to address all of the symptoms of PWS in one fell swoop. We've yet to know that for sure, but at least the potential is there. This is a very young, enthusiastic researcher who has done a lot of work and was very productive in year 1 of the project. And she is in a larger lab that is one of the leaders in using CRISPR technology to activate genes. So they have done a tremendous amount of work sort of advancing the general field of how you use these molecules to the best effect. So this project will lay the groundwork towards therapeutic development of a one-time transient epigenetic therapy with the goal of stably reactivating the PWS genes for the lifetime of the patient. The next project is from the Dykens group at Vanderbilt University. This is a highly experienced group, have done just a ton of PWS research, and they recognize, as many of us do, that the hyperphagia measures that are currently used in PWS clinical trials, while they are important and they have proven useful, there's a lot of room for improvement as well. And particularly the FDA is very interested in, perspective of the person themselves and not so much the caregiver's perspective. So any measure that we can do that is based on the perception of the person that is receiving the drug is favored by the FDA. We know that the HQCT, the Hyperphagia Questionnaire for Clinical Trials, can be vulnerable to informant bias, meaning the caregiver, the caregiver's perspective. And again, the FDA is interested in having the perspective of the person who's receiving the therapy. So this is a difficult task in PWS. We all recognize that, but I think this group is so knowledgeable about PWS and sort of the personality of individuals with PWS that they are going to try to develop a self-report of hyperphagia for individuals with PWS. So we're excited about this, again, because of the focus on the perspective of the individual who is experiencing the hyperphagia. This method might circumvent the specific approach that they're using because they have such knowledge of PWS and some of the cognitive difficulties we see in PWS. They're going to adapt it to the challenges of asking a person with PWS about their, about their hunger levels. So it may offer a new approach to this challenging problem. And this is a very experienced group and probably the only one who, could really address this directly with the individuals with PWS. And I think finally, we have a project from Casey Bedard and her group in Chicago, looking at applied behavioral analysis in PWS. We know anecdotally, a lot of families has found this very useful, but there's not too much literature that you can, sort of take to your insurance company or take to your doctor and say, yes, we need ABA therapy for a child. And so this project is based on a pilot study that they did that is really educating parents about how to use ABA effectively for their children with PWS. And that was a very effective pilot. And so in this study, they'll be expanding that effort and gaining additional evidence that will be helpful in, solidifying that ABA therapy is really beneficial to those with PWS. So we're excited about the study. This group is very enthusiastic. They're very efficient in getting these studies done. We know that caregiver support is a priority, and educating families so that they can, use this ABA approach at home is really critical. This study is it's research-based, so it's looking at what are the most effective ways to do that. And the long-term goal is to demonstrate the effectiveness of this PWS Smart Start and the value of ABA therapy in PWS. So I'll skip to the end, and Susan, I guess we'll take questions from everybody. Just wanted to, say what can we expect as we move into 2023? I think there's a tremendous amount of momentum in the field, and I'm very enthusiastic about that. We will be making a few more announcements. Believe it or not, this is not all the grants that we've funded. There's a few that have paperwork pending. So look for some additional grants to come out between now and the end of the year. We also see, based on the scientific conference, that there's going to be some changes in our understanding of PWS biology that will really, I think, impact the field in general and, make scientists think differently about ways to treat PWS. As I said, we really have a really nice critical mass of scientists that are now working in this area and exchanging ideas and building on each other's work. There'll be new developments on the clinical trial front. We'll see Soleno's data, hopefully in, the first half of next year. And we're hopeful that we'll push them towards being able to get an FDA approval for DCCR for our loved ones with PWS, which would just be fantastic. We're interested in seeing Harmony's full data set and their next steps, and then the initial outcomes of some of the newer drugs that are being tested in PWS. We're also going to be launching the study of vagus nerve stimulation to reduce temper outbursts in PWS. There's a video about that in the 2022 conference YouTube channel that you might want, if you're interested in that, you may want to learn more about that. Please look for additional announcements. We're really excited about this non-drug intervention that might help behavior in our older kids and young adults with PWS. We've got new initiatives from the PWS Click group to improve care, analysis of the path for PWS study to help us understand what serious medical events are occurring in people with PWS. And finally, continued activity of the Clinical Trial Consortium to improve the clinical trial experience and increase the efficiency of moving towards drug approvals in PWS. So that is all I have. I realize it's close to the end of time. If there are some questions, maybe we can stay over a little bit if people have a moment.
So I love your statement as that's all we have. We just went through an hour's worth of material. Clearly FPWR is doing a lot. There's and the scope of these projects, from the basic science, genetic therapy to behavior therapy, cognition. It really makes me excited to see just the breadth of work that that we're able to fund, and really all of that funding is made possible by the folks who have joined us today to learn about this research. So a huge thank you to everyone in our community who are doing fundraisers, donating, participating in the registry, really making all of this work possible. We are up against the hour, so unfortunately, we just don't have time to answer questions. However, Theresa has provided her email address here on her slide, theresa.strong@fpwr.org. So I'd certainly encourage any of you who feel that your question was not answered today, please feel free to reach out to Theresa directly. And we will be looking forward to seeing all of you again in 6 months when we have our next research update. Have a great and wonderful day, all. Bye-bye.